Hello, everyone. Welcome to our virtual online biology labs. So today is the first lab where we'll be working on microscopes. The goal with these videos are going to be to guide you through all the steps that you would have been doing if you were here in person, in case you need to quarantine or do miss a lab this semester. But ultimately, you are still responsible for knowing all the vocabulary, terminology, and completing all the exercises in each of these labs. As you will be working with the microscopes throughout the semester, you are expected to wear proper attire during microscope operation. This means that, of course, you will be masked. And additionally, to help prevent you from accidentally putting your eye directly against the eyepieces, we have face shields. So you'll see me with a face shield when working with the microscope. So here we have our instrument. It's been carried from one of the cabinets. It still has its dust cover on. When I was carrying it, you should always grab a hold of one of the arms and the base. So carrying it with two hands, as this is a fairly heavy piece of equipment. When you return to your desk with the microscope, you can go ahead and remove the dust cover for now. And the microscope will be placed in this orientation. In order to visualize it at your seat, you're going to want to twist the prism housing and the eyepieces gently around. So that way you can sit comfortably in a chair and look. Now for the other components of the microscope, you have the arm that you will be holding the microscope by. You have the nose piece with the different objective lenses. This is a rotating piece of equipment. Whenever you return it to the cabinet, you need to have it on the lowest objective, meaning that is the furthest away from the stage for storage, so that way you'd never bump anything against it on accident. You can see that at some of the higher magnifications, like at the 1000x, it gets much closer to the stage. You do not want to be storing it that way. So we always stash it with the lowest magnification. Below the objective lenses and the nose piece, you have the stage with a mechanical clip. The slide will fit in such a way with the mechanical clip, so that way nothing is done by force. Does not go under anything, it sits on top. Beneath the slide, you will see the iris diaphragm and the substage condenser, a knob that can move left and right here. This will control the size of the iris. And then you have the actual lamp itself, controlled by a switch on the back to turn on the light, with intensity being controlled by a dial on the left here. In terms of movement of the stage, you can move in the up, down, or Y direction using the top dial on this control to get the sample above the iris. You can move it in the X direction or left, right using the lower knob. And you can move the stage up and down using the coarse, the big dial, and the fine adjustments. These are for focus. These are for location. You should always begin with your microscope at the lowest objective. So you will get the sample lined up over the light. Then you will look through the eyepiece, as I am here now. And then in order to get it into focus, on the lowest magnification, the 4x, that then goes up through the prism housing and gets amplified an additional 10x. 4x times 10x is 40x for your magnification here, total magnification through the eyepieces. you're going to be using the coarse adjustment. So in order to get this sample into focus, I'm going to crank it till the sample comes into focus. It might take a little while. I will move it left and right as necessary. It seems pretty bright to me here, so I can reduce the light so that way I'm not staring at a very bright light the whole time. And then once the sample is in focus at 4x, here you can take an opportunity to take an image, or using your cell phone, or if you want to go to a higher magnification, once it's in focus at the 4x, you can then rotate the nose piece to bring down the next higher magnification of 10x. So now we're looking through a 10x and a 10x in the eyepiece. Magnification is multiplicative. So we're at 100x magnification. And if your sample was in focus, it should be close to focus here. If you need to adjust it, now that you are at a higher magnification, you will never use the coarse adjustment. You will only use the fine adjustment. Get that into focus, and you can see the images produced on this in just a second. All right, so here we have a series of microscopy images. We'll first be looking at all of the images at 400x, so hopefully you yourself are able to use the microscope to be able to find our different cells at 400x. This, of course, requires the ability to focus first at 40x, then 100x, then 400x. In each of these slides, I'd recommend that you maybe even zoom on some of these images so that way you can identify all of your different cells in each of the 4 easily identifiable stages of mitosis: prophase, metaphase, anaphase, and telophase. Cells in prophase will be characterized by having these very distinct condensing chromosomes. Cells in metaphase, I'm not seeing one right here on this slide, but we may find one here on the next one, will be characterized by having all of their chromosomes in a neat bundle, in the middle, lined up at metaphase. Cells in anaphase will be separating. These look like they're a little bit later in telophase, or separated. And then, of course, telophase are completely separated with the beginnings of a cell plate in between. Now, in addition to looking at these from quite far away, you could use the manual zoom on your phone to be able to see greater detail of a cell in metaphase. Look at all those centromeres lined up along the metaphase plate. Cells in anaphase, neatly separated. Perhaps some earlier prophase, later prophase. And then a beautiful cell here in telophase, with the cell plate almost extending all the way across. So just like we were looking at our cells in plants in mitosis, we're looking at whitefish mitosis here at 400x. Again, you're going to want to be able to use the microscope to identify these different cells at different magnifications, though your magnification that you're going to want to be using is 400x. Apologies. This is zoomed out. Again, perhaps you can identify some cells from far away. Another zoomed out version. This is actually what you'd see under the microscope. So from this perspective, it's useful. But again, you can use the zoom
on this video yourself to be able to see some cells in greater detail, and a third image at 400x. And here again, you'll have some closer images based on camera zoom, where you'll again be looking for our cells in prophase, with chromatin beginning to condense, perhaps in metaphase, where things are just lining up, or in anaphase, as they're dividing, or here beautifully in telophase, where you can see the pinching from the outside in beginning to occur with our two bundles of nuclei starting to decondense. This would be very late telophase with the pinch all the way down done and the chromosomes decondensing into lighter stained chromatin. And again, with camera zoom, we see closer up images in late anaphase, potentially soon going to be beginning telophase, some early prophase, so hopefully you can spend some time looking at these different images to get yourself accustomed with looking for cells in all of our different phases in both plants and animals. I would note that you should certainly go back through this video to identify the differences that you see between plants and animals, most notably surrounding cytokinesis.
Hey everyone! So we've now gone over our mitosis stages under a microscope. So we've looked at both plant in the onion root mitosis as well as our animal whitefish mitosis, and hopefully you get a feel for the way in which the different phases look under a microscope. That's really what I want you to be paying attention to. Now we're going to take the opportunity to talk about meiosis, because that is fundamentally what so much of our labs, so much of our lecture, at least this first half of the semester, is going to be about. Where we're talking about the division of chromosomes, that setup, and how that ultimately relates to transmission genetics. So the way in which we're going to do this is by looking at pipe cleaner chromosomes that we've color-coded. The color coding is as follows. Derived from father as blue, derived from mother as pink, with chromosome 1 being yellow, derived from father as blue, derived from mother as pink, with green being chromosome 2. In order to be a diploid organism, you have to have two sets of chromosome for each chromosome. One from mom, one from dad for chromosome 1, one from mom, one from dad for chromosome 2. You'll note that these are not forming up as X's. These are each individual strands of DNA. Let's walk through the entire process from interphase through meiosis in our model plant cell. So here we have our 4 individual pieces of DNA in G1. You'll note that in G1, the cell is just going to be gathering nutrients, so things are coming in, resources are being gathered, you've got organelles being copied. In S phase, you do DNA replication for each of these. So from our template of dad's chromosome 2, we have an extra strand, a second copy of this. From our template of dad's chromosome 1, we copy an extra copy of it. Mom's chromosome 1 and mom's chromosome 2. All separate strands in S phase. In G2, the cell is again inflating, gathering its resources, and getting ready for that process of meiosis. Once we enter meiosis, the first thing that begins to occur is this chromatin that's unwound and spread around the cell. You remember it from looking at the cells as being light purple. It starts to condense into visible purple strands. And homologous fragments start to pair up and entangle. At the same time that they're all coalescing together, the nuclear envelope is disappearing, the nucleolus is disappearing, and our spindle fiber is forming on each of our two sides. That will conclude prophase 1. In prometaphase 1, you then have the kinetochores forming at each of these chromosomes in order to allow them to move. Now, if we were lining up for mitosis, they would line up above each other. 1 deep, 4 long. However, in meiosis, you are going to be pairing homologous chromosomes. So, green with green, green or yellow with yellow, and it is variable as to whether the pinks will all be on one side with blues on the other, or some blue matching with some pink. There is your law of independent assortment. These chromosomes don't know whether they're derived from mom or dad, so they're lining up in such a way that's completely random. At this point, during prometaphase 2 prophase and actually G1 and S phase, during this you have recombination occurring. So while these are paired and while you have these homologous fragments together, the chiasmata will form, and you will get occasional breaks here. We won't be illustrating this in this diagram, but know that you can get breaks, and on average you're getting 1 recombination event per chromosome arm. So you'd get 1 here between these two, 1 here somewhere between these two, 1 here somewhere between these two, and 1 here somewhere between these two, on average. 1 per chromosome arm per meiotic event. Now that the kinetochores have moved everything to the middle, and our chromosomes are lined up 2 deep and 2 wide, different from mitosis, we've now reached metaphase 1, where our homologous chromosomes will split in anaphase. So now you've got the situation where this cell has both fragments of DNA from dad, from chromosome 2, both fragments of DNA from chromosome 1 from mom, and this cell gets 2 fragments from mom from chromosome 2 and 2 fragments of dad's chromosome 1. So whereas you would expect both of these products at the end of mitosis to be exactly identical, you now have genetic separation, law of segregation. These different chromosomes of different parentage are splitting apart from one another. During telophase, everything that happened in prophase 1 gets undone, so that means these kind of start to decondense, deorganize. You've got the nuclear envelope forming again. The nucleolus is temporarily forming, though it'll go away in a second, because the moment these two have split and there is a break down the middle, you then immediately proceed to prophase 2. In prophase 2, everything wraps up. Prometaphase 2, kinetochores will form, but now, in the second bout, I'm going to pretend that this is now in its own separate cell. In the second bout, you are lining chromosomes up 1 deep. So that way they're no longer pairing with homologous chromosomes, but each of their fragments are lined up on the metaphase plate. Prophase, they've organized. Prometaphase, kinetochores have formed. Metaphase, they've lined up. Anaphase, these will break apart. These will break apart. So that way now you have unique genetic combination here, unique genetic combination in separate cell here, unique genetic combination in separate cell here, and separate cell here. So you've actually got division between each of these, one cell with its own combination, another cell with its own combination, and a fourth and final cell with its own recombination. Now, here you can tell that these two are identical, but note that in reality, recombination would have occurred between these pipe cleaners, and same here. And in that way, meiosis is able to result in 4 unique gametes. Cool.
Hi there, Rowdy.
But rather than being 1 copy from mom, 1 copy from dad, these are 2 from dad. These are 2 from mom. Now, when they lined up, in this instance, we had dad's 2 with mom's 1, mom's 2 with dad's 1. By the law of independent assortment, you had a 50-50 chance as to whether this was all mom on one side, all dad on the other, or a pairing. In this way, you introduce our first randomness, the genetic mixability that occurs during meiosis that we do not get during mitosis. So, for interest's sake, we'll keep these separating as such. Now you've got this cell and this cell independently going through prophase 2, where they're winding up, again, getting ready to move. Prometaphase 2, where they maybe were not lined up in the middle, they're now moving directly to the middle. Metaphase 2, very different from metaphase 1, rather than lining up adjacent to one another. They again are lined up vertically, 1 deep, 2 tall in this case. In anaphase 2, you're going to be separating the sister chromatids in each of these separate cells, like this. Actually, to be accurate, there we go. And telophase 2, you will form all 4 of your gametes. Now you will notice that because of recombination, this gamete is unique from this gamete, is unique from this gamete, is unique from this gamete. 4 genetically unique gametes thanks to the law of segregation, recombination, and the law of independent assortment.